students. So today we are in week 2, day 2 of the week 2, and in the previous class we have looked at the different sequencing technologies, mostly the first generation ones. So in the today's class what we will do is we will look at the next generation sequencing technology, which has even come after the Illumina, and which is called long read sequencing, and we will do a quick recap of what we did earlier, right, and try to connect with the today's class. Okay, so we have already discussed this previously, that DNA sequencing is very important, and RNA is also can be sequenced similarly by converting into DNA, right. So and the sequencing technologies have taken their own time to evolve since the discovery of the Sanger sequencing technology. Okay, and needless to mention that they have a lot of applications like pharmacogenomics, forensics, right, our health. Today you can go for any SNP based disease detection, and so many tests are available in the market. So in the last class what we did was we did up to this part, that what are the the first generation sequencing technology and the second generation sequencing technology. Okay, so we discussed Sanger and Illumina, but there are some more second generation sequencing technologies which came before before Illumina technology, which we did not do in the last class, and today we will touch those technologies and see that what were their strengths and weaknesses, and mainly we will focus on the third generation sequencing technology in today's class. So the first generation sequencing technology was the Sanger DNA sequencing, which was the chain termination method, where we used to have the 4 normal nucleotides, one nucleotide, basically 4 tubes with 4 normal nucleotides and one nucleotide which have a OH group terminator, or you can say the OH group is modified so that the DNA polymerase cannot extend that DNA further. Okay, so so that means it required 4 nucleotides and one modified nucleotide, a polymerase, okay, and primer, right. So and the strengths were that it can sequence very long, okay, and it is free of errors, okay, longer reads, low error rates. So that is why it is still in practice, but it has a very low throughput and very high cost, so and very tedious method for larger genomes. So this method is not of a choice, and basically what this technology is offered by the ABI like 37330 and several other sequencers which came later. Okay, so 4 tubes, okay, with the all the 4 nucleotides in all the 4 tubes, and one modified nucleotide where the OH group is modified so that the chain cannot be extended, and that is why it is called the chain termination method. Okay, and the long read is still a advantage from this method. So the problem with the previous one was that it cannot be used for larger genomes, and that is why we have the next generation sequencing, such as Illumina, Ion Torrent, and basically started with Roche. Okay, and the advantage is that it is massively parallel. It is not chain termination but sequencing biosynthesis. That means you will pause for for some time, remove that OH group blocker, and then continue further. Okay, and this way you can read that what nucleotides have been incorporated. Okay, so that was the Illumina technology. So it is very fast, high throughput, low cost and low error rate, which are basically the plus points of this, whereas the short read and basically still it required some kind of PCR or clonal amplification are its weaknesses. Okay, so this was Illumina that we have already done earlier, but there are few other methods that came for the second generation sequencing. Okay, one is pyro sequencing. So if you see that basically whenever a nucleotide is inserted, okay, so one pyrophosphate group is released. Okay, so whenever one DNTP, it could be any nucleotide A, T, G or C. Okay, that is why it is called pyro sequencing. So whenever it is inserted into the sequence, okay, the reaction releases one phosphate. Okay, so now you by using this phosphate, if we have APS plus this phosphate group, by using the enzyme sulfurylase, it can convert that into ATP. So that means for every nucleotide that is being inserted, okay, we can detect that by generation of an ATP. Okay. Now how will we actually detect that ATP? So basically enzyme called luciferase, okay, which can generate light, but it needs ATP for that. Okay. So when ATP is released, this ATP, okay, this luciferase this uses this ATP and produces light. Okay, and then light is a signal that nucleotide has been inserted. Okay, but what nucleotide has been inserted, we don't know. So that is why, like unlike the Illumina method where all the 4 nucleotides were there, here we gave nucleotide one by one. Okay, so we gave one nucleotide, and if it gets inserted, it will release a phosphate, and we know that we provided A. So that means if light is produced, that means A was used or A was inserted, and this way we can detect, okay, or the 4 nucleotides. So basically this is also an example of sequencing by synthesis approach using an optical system. Okay, it requires clonal amplification because you need to have sufficient quantity of DNA for production of sufficient amount of light that you can detect. Okay, it releases a pyrophosphate by, okay, which is subsequently used to generate the light. Okay, it is also massively parallel, and up to 3 lakh samples can be done in one point of time. Error rate is not much, it is 1%. Okay, although the newer technologies have much lesser error rate. Okay, so the advantage is that it can also do very long sequence, like up to 1 kilo base pair, like the Sanger one it can do, whereas Illumina can do only 150 base pair or 300, 250 or 300 base pair. Okay, relatively fast, but actually it is very expensive because you have to use a lot of reagents, you have to do washing, right, and that leads to wastage of reagents as well as the enzymes. So that is why this technology was came after Sanger, stood there for some time, and later on basically now it is not used. Okay. Then came Illumina method, which was we discussed in the last class, where you prepare for every DNA molecule, you put adapters at both end, okay, and then do the clonal amplification. These adapter will go and bind here, okay, and then basically this will form a bridge, and it will be read in this way from both sides. And basically what the objective of each of these zones is that in this zone basically one DNA strand is being amplified into multiple copies, okay, and then basically this is called one cluster, this is called one cluster, and each of this cluster, whenever a nucleotide is basically inserted over there, okay, based on the nucleotide which is inserted, okay, because all 4 nucleotides have 4 different signals, okay, you will get a signal from this region which will be actually be used like this. So basically if you see different dots here, these are the different clusters which are producing a signal. So that means all the nucleotides in this region, okay, will be producing one signal each, okay, of the same nucleotide, and that will amplify the signal, and which will be detected as fluorescence here, okay, which is then captured by the camera. Okay, so this is a massively parallel technology, very less error rate, but shorter reads, okay, very cost effective, very cheap as compared to the other methods. Okay, so this is Illumina. Okay, and now they have Illumina is offering systems for all kinds of need. For example, iSeq, which is very small, okay, can give only 1.2 GB of data. Okay, that means good for one small genome like a bacterial genome. Okay, and shorter reads 2 into 150 base pair. Okay, so basically for resequencing purposes and for clinical applications. Then MiniSeq, which can be used for little longer data output, little larger, like up to 7.5 GB, which could be a single metagenome or smaller sequencing projects, and 2 into 150 base pair read length, right. Then NextSeq, which can produce up to 120 GB of data, which is good enough for a good genome resequencing, or even new sequencing, because genome sizes are typically like 5 MB to 3 billion base pairs if you see the range from bacteria to say humans. okay, and 2 into 150 it can produce, and relatively free of error. So this is one of the method of choice for smaller labs. Then MySeq, okay, advantage is that it can produce longer reads up to 300 base pair, and but data output is like 15 GB, okay, again for smaller labs. And then for the big sequencing centers or for commercial purposes, HiSeq 2500 and NUX and NovoSeq 6000 are the two sequencers which can produce, you know, up to 6000 GB or basically 6 TB of data in one run, and 2 into 250 base pair you know, reads, and very cheap compared to all the other systems. Okay. Now in the second generation sequencing, we have seen two methods. One is pyrosequencing, where a phosphate group, pyrophosphate group is released, okay, and which is later on converted into a light signal, okay. Then basically next one is sequencing by synthesis, where you use a reversible terminator, and for every base pair you read that, you know, how what is being inserted, okay, after the clone and amplification on the flow cell. And there is another technology called ion semiconductor sequencing. So in every base pair that is inserted, okay, in the chain, whenever a nucleotide is inserted, basically it releases a proton, okay, and which can change the pH of the system. So this principle is being you know, exploited here, that for the insertion of every nucleotide, the proton that is released, okay, can be captured and read, okay, based on the change in pH in the system, and it can tell that, okay, the reaction was successful, that means one nucleotide was inserted, okay. So basically again this is a sequencing by synthesis method, and it's non-optical but uses a very smart and you know, very sensitive pH sensor, right. So hydrolysis principle is that the hydrolysis of the nucleotide releases a proton, okay, H plus, okay, which changes the shift or changes the pH of the surrounding medium, okay. One proton may not change, that is why you need you know, a lot of protons, that means a number of copies of the same DNA, okay, which is detected by the sensor. So this proton change or the pH change is detected by the sensor, okay, leads to converted into voltage and leads to detection of the nucleotide, okay. So there is no optical system or camera is required, okay, longer read length, faster, and error rate is slightly more as compared to Illumina, right. But it has some issues like homopolymer sequences, when for example the sequence contain A, okay, or T, or G, okay. So all 4 A's will get incorporated at once, and you will not know whether it was one A or multiple A's that got inserted, okay. So homopolymer sequences are difficult here, and the reagent cost is high in case of ion torrent sequencing, okay. So this is the principle that okay, for A, T, G and C, you will know that if you are giving C and there is a change in the pH, that means C was the nucleotide that was supposed to be inserted, okay. And again the read length is 100 to 300 base pair. This method is still existing today, not so popular like Illumina, but still it is one of the method that is used for large scale sequencing. Okay, so now in the second generation sequencing, we have seen that the read lengths are smaller, okay. They require basically a polymerase, right, and a reaction which can be detected in the form of light or fluorescence or proton, okay, which is actually captured by the system, and you know that a nucleotide is inserted, okay. Now the third generation sequencing technologies are different than the first, okay, in the sense that they will not need larger amounts of or copies of the same DNA. So basically they are amplification free, okay. Second, they can produce long reads, okay, because of their, you know, technology that we will discuss, okay. Though the error rate is little higher at this point of time, but in the future or basically in the upcoming versions it is being reduced, okay. And there are multiple methods like SMRT or PacBio, basically, right, both are similar, and Oxford Nanopore technology, which we will discuss today, which is based on Nanopore, okay, and synthetic link reads, which we will also discuss today, right. And there is one more which is short Illumina reads with along with 10x genomics. The combination of two can be used, but 10x genomics is now no more supporting the link read technology. So very soon I think they will come up in another version of that, but till then we can discuss the principle, but right now it is not in use. Okay, so PacBio is one sequencer which can do long read sequencing. Menine, Gridine are the two nanopore devices, and Chromium 10x along with Illumina can do the long read sequencing. Okay, so first briefly discuss what is link reads. Okay, so basically link reads are prepared by the Chromium instrument which is basically by the 10x genomics. 10x means like thousands of something, okay. So 10x means large scale, okay. So basically what it requires is a DNA which is fragmented into smaller fragments, and then each fragment is barcoded separately, so that we can trace back that the shorter reads that is generated from that fragment, how they can be arranged together to form the complete link reads, okay, or the complete contiguous read, okay. So basically it requires barcodes, right, a small benchtop instrument, right, and it can provide long range information from the short read, we will see in the next slide, right, and a advanced microfluidics platform, because we are dealing with multiple fragments and their barcoding, okay. So okay, so basically the multifluidics enable the DNA fragment to attach to bead, okay, like this. This is a bead and this is the DNA, right, and then the unique barcode for every read is generated from the individual molecule, okay, which is then sequenced. So if you look at this image, you can this becomes clear. So basically this is one intact DNA, right, which is now broken into three pieces: gene 1, gene 2 and fragment DNA fragment 2 that contains part of the two genes. I am just writing gene for convenience, otherwise it is basically two different regions in the DNA. So basically these three fragments are generated. For example, because we are doing random fragmentation of the DNA, we don't know what piece is what, right, but these are sufficiently longer pieces. Now each of these long piece, okay, is labeled separately, okay. You can see green and brownish and yellow, three different barcodes are there for each piece, okay. Now the fragment that are generated from each of these piece will also be, okay, labeled by the same barcode, okay, and then you are sequencing them. So when you are sequencing them, you will have the sequence as well as their barcodes, okay. So now when we sequence, we know that, okay, these green barcodes are coming from the same DNA fragments, the yellow ones are coming from another DNA fragments, and then we can use this information when we are assembling them, right, into the assembled genome. So basically we will look for all the pieces with green tags and this time computationally, and assemble them into longer pieces, which is and that is how we use the principle of link reads in 10x genomics, and the sequencing is actually done by the Illumina instrument, okay. Now come to the another very important and common DNA sequencing technology of the third generation, which is nanopore sequencing. So nanopore, that means there is a small nano dimension pore which you can see here, okay, right. So basically this pore can allow a single stranded DNA molecule to pass through, okay, and okay. So basically it will not require a polymerase, right, but what it will require is that basically through this pore, when the DNA molecule is passing, right, the current is also passing, okay, ions, right. So basically okay, so basically you can imagine that there is a small pore in which the current is passing, okay. So you can detect the ions are passing, and that is can be converted into a small current, okay. But when I block the flow of those ions, okay, so that will lead to change in current that I will detect at the end, okay. And that change in current is because of A will occupy different size, T will occupy different size, G and C, Okay, so basically they will differentially disrupt the current through that nanopore. So this technology is offered by Oxford Nanopore. Okay, so basically it can do up to 400 bases per second, and these protein pores are embedded in a membrane. Okay, the disruption in the ionic current is actually measured after passing of each nucleotide, because each of the 4 nucleotide is of a different shape, and they will differentially block the flow of ions through that pore. Okay, and that the change in the flow of ions will lead to change in the current. Okay, and that we are detecting to see that what nucleotide was there. Okay, so it is real time data streaming, because we know that what is the pore, right, and what molecule is going through that, because okay, so we can detect while the sequencing is going on that what nucleotide was present or being detected at that point of time. There is no requirement for PCR, because basically you are using a single strand, or okay, double stranded DNA converted into a single strand which is passing through. Okay, so there is no polymerase, no nucleotides, right, involved. It is just the change in the ion flow which is recorded, right. So it saves in terms of the expensive consumables like the enzyme or the nucleotides, right. Sample preparation is quick, and it gives you data in the commonly available formats, and sequences are MinION, GridION and Promethean, which we will see in the next slide, right. Okay, so these are the different sequencers. So this is a very popular sequencing MinION. So you can convert you can attach this MinION to your laptop using a USB. So it is like very small, this size, right, and basically it has a flow cell, okay, which is like a glass slide size, and you can do sequencing anywhere you are, right. The other one, so basically in this setup, in this MinION device, at least one laptop is required, but they have another like smaller 8 inches kind of a small pad kind of thing, which does not require a laptop, and it has about 1 TB SSD and 1 GB RAM. Okay, and basically 8 GB RAM, and it can do the sequencing on the runtime, and also can do the base calling. So basically no laptop is required for this kind of device, and there are bigger versions also like GridION and Promethean, okay, which is for the bigger labs, and okay, we will see in the next slide that how they are different. So MinION can give up to 30 GB of data. So MinION has 2 versions, MK1C and MK1B. This is USB type, and this is independent type. It does not require a laptop, right, and this can also give MinION, both can give about 30 GB of data, right. And the bigger ones like MK1 and GridION and Promethean, they can give up to 150 GB or even some terabytes of data, okay, and which are for bigger centers. But actually this MinION and both devices are not very expensive, so okay, and they can be easily bought in labs, and that is why they can lead to more affordable and faster sequencing. Okay, so this is another picture of these. So like this is a device, this is the typical flow cell, where you load the sample at this point, right, and okay, so basically this device remains same. You just change this flow cell, which is which contains the nanopore, and after it is exhausted or used, they can be washed like once, and till their pores are alive. Otherwise, once the pores are not there, there is no need for reusing them, and then you throw that away and keep this device, and this is now thrown away and use the another one. Okay, so the technology is that basically there is a pore, nanopore. Okay, there is a helicase enzyme, because the DNA is double stranded, so it has to be converted into a single strand. So this helicase converts the DNA into a single strand. Now the single stranded DNA passes through this membrane, okay, and at the same time the ionic current, or basically the ions, were also passing through this membrane. Okay, so now this DNA, if you see, these beads contain A, T, G, C, 4 different type of nucleotides, and this nucleotide will occupy a different region or spatial occupancy in this pore, and this will differentially block the flow of this ion through this pore. Okay, and that differential blockage of flow of ions will lead to differential current. So if you see here, so basically this is the DNA molecule, double helix, okay. One helicase actually unfolds it and one strand goes in, okay. In the second step, basically this is going through this, so this is the adapter molecule that is basically keeping them there, right, and they are passing through this pore, okay. And so this is a nucleotide which is occupying some space, and these are the ions which are going through. So you can see that if there is no such obstacle, there will be more ions which will be flowing through, but due to this obstacle, basically it will lead to reduced ion flow, which will lead to reduced current at which is detected by the device, okay. So this is the and this way, like, you can detect whether A has passed or T or G or C, and that is why this technology is very efficient, okay. So this is like another picture from this. These are different nucleotides blocking, because of the different shapes, they are blocking the pore differentially, okay, and leading to change in the ions which are flowing through, and this change is then detected in the form of current. A, T, G and C will produce different kinds of current, okay, right. So this is a very good technology, and now the error rate is about you can say 10%, but it does not require too many consumables, and much cheaper compared to the Illumina, and does not require a typical assembly process that is very time consuming and wasteful, and also depends upon your logic and the further processing, which is not there. So it is producing a very long read, though it has errors, but if you have multiple reads from the same region, okay. So for example, if this place was an error, so I can check the same position in the another read or the other read and the other read. So if I say that, okay, this is A, but in the other reads it was T, T, T and T, so I can say that, okay, this was T. So this way error correction can be done, or this can also be done by mapping the shorter reads by Illumina technology to this long read, which is kind of a hybrid sequencing approach, okay, because Illumina technology is very good for in terms of accuracy, and if I map another smaller read in this region and I know that T was there, so I can know that I can correct the error. So basically contiguity is much better in case of the nanopore reads, okay. So basically what happens is, if just a small overview of the genome pipeline for nanopore, you detect that what is the signal, okay, in terms of by the device, okay, and that data is used to do the base calling, that what base was present, it was A, T or G and C, and that converts into a long read. Now that reads that you get, you try to find an overlap between the two reads, okay, and connect till basically there are some reads which are overlapping, okay. This is called assembly, and once you have assembled this genome, right, so basically you keep on assembling till there are no gaps or a very less number of gaps, or you don't find another overlap, okay. And then on this draft assembly, basically you can do again the read mapping to correct it, or do the Illumina read mapping to correct this assembly, okay, and then followed by polishing. So all these steps like assembly and polishing we will also be doing in the next classes, okay. So basically this is a good technology that produces very long reads. It is much less expensive, instrument is also not very expensive, right. And so basically in this class we looked at the newer and older sequencing technologies, a comparative of that, okay. How nanopore is different from Sanger or Illumina or ion torrent or pyro sequencing, right. We also looked at the concept of link read sequencing, where you differentially label or barcode each fragment, and then fragment of fragments, you can use that for assembly, okay. So this is good for this class, and in the next part of this class we will look at the other third generation sequencing technologies, right. Okay, thank you.